The two questions I asked are important as they provide a brief introduction into inserting targeted genes into plasmids for the purpose of cloning and perhaps the creation of a transgenic plant like golden rice. Problem 1a requires you to know the function of the different items on the vectors, while problem 1b requires you to know how restriction enzymes work. The reason I did not ask directly how to say input the PYS gene from either daffodil or maize DNA into a vector, as is done in the creation of golden rice, is because that process is far more advanced. Not only are two different restriction enzymes being utilized, either BAMHI or Hindi2, or BAMHI and ASP718, depending on which vector you're using for the daffodil PSY gene, but specific promoters must also be added, and polyadenylation must also happen. The use of two different restriction enzymes is not ideal and was never even gone over in class, so I decided to use a more basic example that still gets the same ideas across. Question 1a wants you to identify which vector will give results that are easier to analyze and requires one know that LAC-Z in the presence of X-gal will create a blue color, allowing color differentiation if insertion of the gene breaks up the LAC-Z gene. Question 1b requires one to look at the plant DNA sequence and plant vector and decide which restriction enzyme works best, which in an ideal situation would be one enzyme that has restriction sites on both sides of the target gene, gray color, and one cut on the LAC-Z gene of the plant vector. This happens to be the restriction enzyme BGL2.